talk about the evolutionary journey of the weird and wonderful Gropioid amphipods. So as you all know, amphipods are found in all aquatic habitats around the world, and they are critical to the foundation of many food webs. They are abundant primary consumers, which recycle nutrients, and perhaps most importantly, they are a primary food source for many marine predators. This means that most amphipods are vulnerable to visual predation. And most amphipods use their highly volatile ways to avoid predators since they lack a thick outer protective layer. And as you all know, the generic amphipod body is divided into 3 primary regions, which form a laterally compressed bean shape in most groups. This bean shape makes them good at quickly wiggling away. It also makes them good at avoiding tight crevices and under rocks or algae. However, there are important groups which have modified this form. Meet the Gropioid amphipods. Gropioids are amphipods which belong to what is currently recognized as the Grophiida infraorder. Unlike their supermobile bean-shaped relatives, Gropioids tightly cling to exposed surfaces. Many live in tubes to protect themselves, while others are free-living with extreme body types. And despite their more exposed lifestyle, they are some of the most morphologically diverse amphipods in the ocean and some of the most ecologically important marine food webs. However, it's unknown how the ancestral Gropioid transitioned into such amazing diversity. The reason for this knowledge gap is because previous research has only compared small numbers of shared genetic or morphological traits. So my research aims to clarify Gropioid relationships and key evolutionary transitions using a much larger genetic data set. In 2003, Myers and Lowry predicted that Gropioid evolution was the result of traits which enabled a series of new amphipod lifestyles. Likewise, my hypothesis is that modern Gropioid diversity is the product of cascading key innovations. So the catalyst for this cascade is silk. Silk spinning has evolved multiple times in arthropods, but the trait is extremely rare amongst crustaceans. The idea is that silk spinning allowed a free-living bean-shaped ancestor to transition to a protected tube and then gain a distinct fitness advantage. And this led to the initial radiation of soft tube dwellers that we see that build tubes out of mud or algae. And tube dwellers that gained elongated antennae could then suspension feed from the protection of the tube without exposed foraging, and this would lead to a second radiation, which would produce the many rigid tube dwellers that we see in areas with high current. And then suspension feeders would benefit from extending further into the water column, and they would also benefit from opportunistic foraging since water current is variable. And by modifying the urosome, the hind periosome could become a rigid grasping base and allow some of these suspension feeders to leave their tube in these high current areas. And this would lead to the final radiation of free-living clingers. And by leaving the protected tube life, many of these taxa might lose the ability to make silk and find alternative ways of avoiding predation, such as funky camouflage or bizarre associations with better defended animals, hence producing the weirdest bodies amongst the Gropioids. So to test this hypothesis of cascading key innovations, I am using a robust phylogenetic framework. Specifically, I'm using a large genetic data set to build a Gropioid phylogeny, and then using this phylogeny to identify traits which precede major changes in lifestyles. I collected Gropioid specimens from biodiversity hotspots along coastal margins, and for transcriptomes, I collected fresh specimens and RNA later, and for target enrichment sequencing, I used preserved specimens and ethanol. And a large part of my sampling took place along the west coast of North America using scuba or intertidal methods, or through collaboration with my colleague Sienna McKim at UC Santa Barbara. I also acquired fresh specimens from Antarctica thanks to a collaboration with Kevin Kosa at the University of Alabama and his research crews on Nathaniel B. Palmer. And to improve sampling of groups that were poorly represented by the fresh specimen collections, I also acquired preserved specimens for target enrichment sequencing. And this included lots along the east coast of Australia and the west and east coast of North America and the Caribbean. So overall, I acquired representatives of each predicted radiation in my hypothesis. For transcriptomes, that's 20 genera from 9 different families, and additional genera within these families for target enrichment sequencing, as well as an entirely additional family, the Cyanidae, or the whale lice, with target enrichment. So with the help of chemicosa at the University of Alabama and Sienna McKim, we extracted RNA from the fresh specimens and built cDNA libraries for transcriptome sequencing, and we used an Illumina NovaSeq to acquire whole transcriptomes for each specimen. I also designed custom baits from these transcriptomes, and then I used these baits to target and sequence specific genes from the preserved specimens. I acquired transcriptomes for outgroup taxa from GenBank or published genomes. And then I chose crustaceans from different evolutionary distances so that the outgroups would cluster together and to prevent long branch attraction with the faster evolving lineages of my in-group. So once I had the reads, I performed the phylogenetic analysis, and without going into too much detail, first I assembled transcriptomes from the RNA, and then generated alignments from the orthologous sequences and built gene trees and a partitioned species tree. Then I used this transcriptome dataset to design baits to target and sequence 135 genes across 3 distantly related Prophyoid taxa that span the breadth of Prophyoid phylogenetic tree space, and then generated alignments from the taxa that had greater than 20% target gene recovery from the enriched data and the transcriptome data, and then used these alignments to build the final species tree with the original transcriptome tree as the backbone constraint. And then I used this final species tree to perform ancestral sequence reconstruction with a custom scriptum R that was written by Sienna McKim and Emily Lau at UC Santa Barbara. And for this presentation, I chose 4 traits related to different lifestyles, and these included elongation of the peduncle of the first antennae, silk spinning systems, a tube structure, and urosomite modification. And Sienna McKim determined the silk spinning systems using for staining and microscopy technique, which I believe she'll be going over in her talk next. So here are my results so far. I built the transcriptome phylogeny from approximately 1,000 shared gene regions, and then I acquired or recovered 56 targeted genes for the final species tree. And on a broad scale, we do see the Prophyoid clade clustering together mostly, and the outgroups falling into their own respective clades. However, when we zoom in on the Prophyoids, we see some patterns that differ from our original hypothesis. We do see a general progression from tube-dwelling to free-living taxa. However, it looks like it's happened multiple times, so there's something funky going on with this transition. If we examine traits with the ancestral state reconstruction, we see some patterns. First, the silk spinning is found in the Prophyoid ancestor, as expected, and it stays with the Prophyoids throughout most of their evolution. If we look at these early Prophyoids, we also see that they are indeed soft tube dwellers with normal length antennae and normal urosomes. We then see an elongation of the peduncle of the first antennae, which enables suspension feeding lifestyles. However, this is where things start to go off the rails. We see some suspension feeders split off into different directions. One lineage stays with a soft tube. Another lineage starts building tube domiciles, or soft tubes within harder external material like algae or shells. And another lineage starts building rigid tubes. And then after this, there's an explosion of different lifestyles and body types, all of which are associated with different ways of living and feeding in the water current. So Notocoma and Dolichias build rigid tubes as mobile or sessile pedestals, respectively. The Steocerotypes use rigid tubes to generate raised fouling masses. The Podicerids, Neoxenotus, and Caprellids ditch the tube life and to cling and extend their bodies. And then the whale lice come out of this and ditch suspension feeding altogether in favor of a clinging symbiotic lifestyle with whales. So as predicted, each of these lifestyle transitions is preceded by a rigid tube-dwelling ancestor that undergoes a urosome modification. However, unlike we predicted, it looks like this has happened on at least 4 different occasions. So 3 lineages fuse or lengthen their urosome to tuck the posterior end and then enhance their clinging. We call these the budded clingers. And then 1 lineage did the opposite and reduced their urosome, and that helped them enhance their clinging. We call these the buttless clingers. So my results partially support the hypothesis of cascading key innovations. We do see that silk spinning catalyzed Prophyoids into soft tubes and then suspension feeding rigid tubes. However, the progression from tube dwelling to free living is not as straightforward as we expected. Instead, it's a complicated story with many transitions. So morphological radiation is related to repeated changes in the urosome of rigid tube-dwelling ancestors. And this means that silk spinning and tube structure are even more important to the evolution of porphyroids than we originally thought. Now, this tree is only the beginning of future work that needs to be done. First, we need to improve target enrichment methods. In this study, I had low target gene recovery because I was limited on funds, and I chose to target more genes across fewer taxa. And this strategy does work with a lot of animal groups, but not with amphipods, because they're just too genetically diverse. So future apes should target genes across more taxa. Second, there needs to be more sampling of intermediate groups, and this tree needs to be rebuilt with alternative models of evolution, which I'm currently doing. Third, the classification of some porphyroid groups needs to be reevaluated. Based on our tree, several families are paraphyletic, and one deep sea family, the Paragamma, doesn't even belong in the porphyroid clade at all. And finally, there's a question of why the porphyroids keep changing the urosome and how they get such extreme body types. And from a morphometric perspective, one hypothesis is that key traits relax the integration of body parts within tagmata, which then allows normally conserved body traits to vary more freely and diversify. So Colin Gross from the Smithsonian is currently looking at this using CT scanning, but it's a work in progress. So in summary, phylogenetic analysis using transcriptomes and target enrichment help clarify many porphyroid relationships. Thus far, we're seeing tree patterns which do support the hypothesis of cascading key innovations, but the progression is much less straightforward than we thought. There needs to be more sampling, but there is a definite promise to use museum specimens to do this, as long as dates are designed appropriately. And most importantly, this tree serves as the foundation for understanding the evolution of silk in amphipods and crustaceans, and this research is currently underway by my colleague Stan McKim, who's presenting next. And so the take home to all this is that silk catalyzed the radiation of a large ecologically important amphipod group, and that a large-scale molecular data set was key to understanding how this trait produced such amazing diversity. So I hope this study shows the value of using next-gen sequencing to understanding new amphipod evolutionary perspectives, and I hope it encourages others to do the same with their understudied amphipod group. I'd like to thank my funding sources and all the people who've helped me with bioinformatics and sampling over the years, including Stan McKim, who's here, and my advisor, Gustav, here today. Thank you very much.